Okay, so welcome back to this next video in which we are discussing ionotropic glutamate receptors. Okay, so we're in the process of discussing long-term potentiation, or LTP, at glutamatergic synapses. Okay, so we've discussed that if you stimulate the presynaptic neuron at a crazy rate, for instance 100 Hz for 3 seconds, what happens is that the excitatory postsynaptic potentials that this generates within the postsynaptic neuron build up. Okay, and this causes a considerable depolarization of the electrical potential difference across the cell membrane, which is enough to remove the magnesium block of the NMDA receptors. Then what happens is the next time the glutamate is released, it's going to activate the NMDA receptors. They're going to open and allow a large calcium current into the postsynaptic cell, and this calcium in the postsynaptic cell is going to trigger changes within the postsynaptic neuron, which are going to lead to long-term potentiation. However, there are still theories about the importance of changes in the presynaptic cell, but I would say that the changes in the postsynaptic cell are more convincing than the presynaptic ones. Well, at least the pathways are more elucidated in the postsynaptic cell. It is most likely the case that there are modifications in both the presynaptic and the postsynaptic cell. However, there is a nice pathway that has been elucidated in the postsynaptic cell, and that's why I'm going to tell you about the postsynaptic cell. Okay, right, so we are going to see how this calcium signal that is coming into the cytoplasm of the postsynaptic cell is going to lead to an increase in the open probability of individual AMPA receptors which have glutamate bound to them. Okay, right, so what's going to happen is at the moment we've got calcium going up within the cytoplasm. Okay, calcium is going to bind to a molecule known as calmodulin, which is also present within the cytoplasm. Okay, so let me show this here. So I'm going to discuss with you now the structure then of calmodulin. Okay, so calmodulin has 2 lobes. Okay, an N lobe and a C lobe. So let's call this the N lobe, and let's call this the C lobe. And the N and the C lobe are connected together by a linker, okay, shown here. Now, both the N lobe and the C lobe both have 2 binding sites for calcium. So here are the 2 calcium binding sites on the C lobe, and here are the 2 calcium binding sites on the N lobe. So in total, 4 calcium ions combined to a calmodulin molecule. Now at the moment our calmodulin molecule doesn't have any calcium bound to it, and this is known as apocalmodulin. So calmodulin with no calcium bound to it is called apocalmodulin, and for short apocalmodulin is often abbreviated to APO, and then the shorthand for calmodulin is CA and then capital M, like so. So capital C, lowercase a, capital M is shorthand for calmodulin. Okay, now apocalmodulin then has this sort of hunched over structure. Okay, so I hope I've kind of shown you how it's sort of bent over. Okay, so it's sort of hunched over like this. There's a curve to its backbone, if you like. Okay, meanwhile this linker polypeptide is actually perfectly linear, so it's just amino acid after amino acid after amino acid. When calcium goes up in the cytoplasm of the postsynaptic neuron, what will happen is 4 calcium ions will bind to the 4 calcium binding sites of the calmodulin protein. Okay, and this is going to transform the structure of apocalmodulin into something known as calcium calmodulin complex. Okay, and what will happen is it will go from having this hunched over structure to this linear structure. Meanwhile, the linker between the 2 lobes will go from being a linear polypeptide to being an alpha helix, like so. Okay, so it's turned from a linear polypeptide to an alpha helix, like so. And this is when 4 calcium ions bound to these 4 calcium binding sites on the calmodulin protein. So here in orange, this will represent the calcium ions, so I'll circle them in orange there. Okay, right. Now, this molecule then, when you have calmodulin with 4 calciums bound to it, this is known as a calcium calmodulin complex. Okay, and for short, calcium calmodulin complexes are often abbreviated to Ca2+ for calcium, and then the abbreviation for calmodulin is capital C, lowercase a, uppercase M again, so calcium cam, like so. Now, the calcium calmodulin complexes activate all sorts of different proteins, but the one we're going to be interested in is something called calcium calmodulin dependent kinase 2. Okay, now there are 4 main different types of calcium calmodulin. Whoops, I've missed off the calmodulin. So insert in here a calmodulin, so calcium calmodulin dependent kinase 2. There are 4 different forms of calcium calmodulin dependent kinase 2, and for short, calcium calmodulin dependent kinase 2 is often called CAM kinase 2. Okay, and that's more often written with capital AM, I think, CAM kinase 2. In fact, no, scrap it again. It has the same naming, I think, as the CAM. I'm trying to decide what's the usual way that people write that. I think that's the usual way that people write it. CAM kinase 2. Okay, the capital K, CAM, didn't look right either. I think that is the usual way that people write it, CAM kinase 2. Okay, but its full name is calcium calmodulin dependent kinase 2. Okay, now there are 4 different forms of calcium calmodulin dependent kinase 2. There is the alpha form, the beta form, the gamma form, and then also the delta form. Now, for our purposes, we're talking about the brain. Now, the 2 most common forms within the brain are the alpha and the beta form. Okay, so CAM kinase 2 alpha and CAM kinase 2 beta, specifically CAM kinase 2 alpha. CAM kinase 2 alpha is the most important one. Beta is the second most important one. These other 2 are a little bit more niche. Okay, right, so what I want to now discuss is the structure of CAM kinase 2 and how it joins together to form these hollow enzymes. Okay, so I'm going to now show you the structure of a single CAM kinase 2, at least a cartoon for the single molecule of CAM kinase 2. And then we're going to discuss how they oligomerize together into sets of 12, basically, and I'll draw you a nice picture for this. Okay, so let's have a look at the structure of a single CAM kinase 2 enzyme then. Okay, so we're starting at the wrong end, really. We're starting at the carboxylic acid terminus, okay, but this is the side that I usually start with. Okay, so close to the carboxylic acid terminus, you have a domain which is called the association domain, okay, or it's also called the hub domain. That's a piece of terminology you will often quite see if you read the literature on CAM kinase 2. The hub domain or the association domain, and this is the portion that's going to be responsible for the oligomerization of the CAM kinase 2 enzymes into these sets of 12, basically. Okay, then what you have is a linker portion, which then has a pseudo-substrate portion coming off here, which is going to be inhibiting the catalytic domain. So let me draw the catalytic domain. Now, CAM kinase 2, as the name suggests, it's a kinase enzyme. It's going to phosphorylate serine and threonine residues within proteins. Okay. Now, at the moment, I'm going to draw the inactive CAM kinase 2. Okay, and in the inactive state, what happens is this catalytic domain over here, which is this archetypally drawn enzyme structure here. So here's the active site. Okay, so this is the portion that will actually catalyze the addition of phosphate groups onto serine and threonine residues. In the inactive form of CAM kinase 2, what's going to happen is another portion of the CAM kinase 2 enzyme will be sitting within the active site of the catalytic domain, blocking its function. Okay, so here is this little portion that is sitting within the active site of CAM kinase 2 and is blocking its function. Okay, and this little portion that's now coloured in purple here, this is what's known as the pseudo-substrate. Okay. Right, so this blocks the catalytic domain from having function in the inactive CAM kinase 2. Okay, and I think we should probably also colour in the association domain down here in turquoise. Right, so this is an individual CAM kinase 2 enzyme. Let's now discuss the form that CAM kinase 2 usually exists in within the cytoplasm of cells, because it doesn't usually exist on its own. Instead, it associates with a whole bunch of buddies. Okay, so basically what's going to happen is it's going to form 2 rings of 6, basically, which are then going to bind one on top of each other. So let me show this. So, how's the best way to do this? So let me just start by drawing a cylinder here. Okay, like so. Now I'll draw this nice and thick, like so. And then I'm going to have 2 rings stacked on top of one another. So I'll divide it into 2 here. Now, we're just considering the top one of these rings at the moment, and this top one of these rings is going to have 6 of these association domains within it. Okay, so it's going to have 6 CAM kinase 2 enzymes contributing their association domains here. Okay, so this portion here is a single association domain from a single CAM kinase 2 enzyme. Okay, and then off this association domain you will then have the rest of the CAM kinase 2 enzyme. Okay, so here comes the linker with the pseudo-substrate coming off here, which will be sticking into the active site of the catalytic domain, which we'll have here. Okay, and this will be the same for all of these association domains. Every single one of them, all 6 of them, will have the rest of the enzymes sticking off the side. Okay, so you'll have a ring of them all sticking off the side, and then on the bottom you've got the same thing again but upside down. So all of these will have their catalytic domains facing upwards. All of the ones on the bottom will have it facing downwards. Okay, so let me draw a picture of this. So here's the pseudo-substrate in purple. Here then is our catalytic domain in blue. Okay, so now let me modify the picture so that I show it on the downside as well. Okay, so you've also got 6 of these making up this bottom ring, and this time they'll be oriented in the opposite direction, like so. Okay, so the catalytic domain here will be pointing off downwards, like so, and then you'll have the pseudo-substrate coming off here and inhibiting the active site, like so. And then you'll have the amino terminus here. Okay, and then we've got the pseudo-substrate sticking in here, and then the catalytic domain here being inhibited. And basically, you have therefore got, in total, 12 CAM kinase 2 enzymes in this huge, great oligomer. Okay, so this will be called a CAM kinase 2 oligomer. Okay, oligo means some, and the refers to many subunit structure, basically. So it's a structure consisting of more than one CAM kinase 2. Right, and the other thing I need to say is that these oligomers of CAM kinase 2, they don't only have to contain one form of CAM kinase 2, they can contain different forms of CAM kinase 2. So for instance, this one here, this could be a CAM kinase 2 alpha, okay, and I think that is the way of writing CAM kinase 2. And this one down here, this could be a CAM kinase 2 beta. Okay, and remember, CAM kinase 2 alpha and CAM kinase 2 beta are the main forms in neurons. Specifically, CAM kinase 2 alpha is the most important of them all. Okay, right, so that's the form that CAM kinase 2 usually exists in within the cell then. We now want to discuss how calcium calmodulin complexes are going to activate these CAM kinase 2 enzymes that are within these huge oligomers of 12 CAM kinase 2 enzymes. Okay, right. So, what's going to happen then is the calcium calmodulin complex is going to come and bind to this pseudo-substrate here, and it's going to cause a change in conformation that will cause the pseudo-substrate to fall out of the catalytic domain, and then the catalytic domain will then have a free active site and can then phosphorylate serine and threonine residues on proteins. Okay, so let's draw a picture of that. So I'll go back now just to drawing a single CAM kinase 2 subunit rather than trying to show this on the entire great oligomer. Okay, but this will be happening within the structure of the oligomer. So here's the association domain, which will still be involved in the oligomer assembly. Okay, and now here is the pseudo substrate, and what has now happened is a CAM kinase 2, a calcium calmodulin complex has come and bound here. Okay, so here is our calcium calmodulin complex, and I'll show it like so. Here are the calciums bound in orange. Okay, and that's caused the pseudo substrate to come away from the catalytic domain, and I'm sorry, I haven't really left enough space here, so the catalytic domain is going to have to be shown rather small. Okay, like so. So let's colour in the different portions here. Here's the pseudo substrate in purple here. Here is the catalytic domain in blue here, like so. Okay, and then here in turquoise, this is the association domain. And now the catalytic domain can go and phosphorylate serine and threonine residues, and basically it's going to now phosphorylate serine and threonine residues within our AMPA receptors. Okay, so if we draw our AMPA receptor here, remember our AMPA receptors consist of these 4 AMPA receptor subunits, okay, and basically all of them have an intracellular portion, and the CAM kinase 2 catalytic domain is going to phosphorylate serine or threonine residues within that intracellular portion. So it's going to phosphorylate a phosphorylation site somewhere on the intracellular domain. It'll do that for all 4 subunits, and this now is going to result in an increase in the open probability of the AMPA receptor when the AMPA receptor is bound to glutamate, so that when glutamate arrives and binds to the AMPA receptor, the actual probability that the AMPA receptor with glutamate bound will be in the open state will be greater. So the AMPA receptors will spend more time in the open state. They'll allow in a greater amount of excitatory postsynaptic current when the glutamate signal is there, and therefore the amount of depolarization of the cell membrane you're going to get will be greater, meaning that the excitatory postsynaptic potential you generate will be greater. Okay, so that's what's known as the induction phase of long-term potentiation. Okay, it's kind of the short-term change that you make postsynaptically to trigger long-term potentiation. You phosphorylate the AMPA receptors via CAM kinase 2 enzymes, and that increases the open probability of the AMPA receptors. Okay, longer term, what you then have is the maintenance phase of long-term potentiation, which is that you increase the actual number of AMPA receptors on the membrane of the postsynaptic cell, so that the postsynaptic cell becomes more sensitive to glutamate, basically. Okay, so those are the postsynaptic changes which you make in long-term potentiation to trigger this increase in the excitatory postsynaptic current that you get for a certain glutamate signal. But it's likely that there are also presynaptic changes as well at the glutamatergic synapse. It's just that the presynaptic changes, the pathways just have not been worked out, basically. Whereas this induction phase, at the very least, this has got a nice clear pathway postsynaptically. Okay, right, even if the maintenance phase hasn't. So, the final little thing then I want to say about NMDA receptors is I want to tell you why they're called NMDA receptors. Well, it's because an agonist for NMDA receptors, which combined to the N2 NMDA receptor subunits, so the GluN2s, which remember are the only subunits which actually bind glutamate. Okay, it combined in place of glutamate and activate them just like glutamate. This is an agonist, basically, and this stands for N-methyl D-aspartate. Okay, so I just would like to show you the structure of this, and luckily I don't need to explain to you what D-aspartate means, because we have discussed both what conjugate bases of carboxylic acids are, so we know what the difference between aspartate and aspartic acid is, and we've also discussed enantiomers of amino acids, so I don't need to discuss what the D means. Okay, so let's now discuss this structure. So it's an amino acid, not a protenergic one because you've got the methyl group coming off it. So we'll draw the alpha carbon here. And then we'll have the amino group here as always. So here's the amino group. Whoops, we're going to have a methyl group coming off the amino group. That's the N-methyl. So let me cross this off. Okay, so here's a nitrogen, and off the nitrogen you'll have both a hydrogen and a methyl. So that's the N-methyl. Now we just need to finish diaspartate. Okay, so remember in the dextrorotatory enantiomer, what you had is you read the corn. You read that in the anti-clockwise direction. Okay, so you want to go around anti-clockwise, which means you're going to want the carboxylic acid group to come out of the page towards you. Okay, so here is the carboxylic acid group, and remember this is the carboxylic acid group of the core amino acid structure, so this isn't the one that needs to be in the conjugate base form. Okay, and now the other group will be going into the page, and sometimes I switch between using that symbol for a bond where it was going like so, where it was parallel lines that grew gradually longer going away, to just showing dashed lines to represent a bond going into the page. Okay, and now we want to put the R group of aspartic acid here. Okay, and this will have a methylene group and then a carboxylic acid group, but of course we're talking about the conjugate base here, so the carboxylic acid group had better be in the deprotonated form. So aspartic acid just has as its R group a 2-carbon carboxylic acid basically. Because we're talking about aspartate, we've got the conjugate base of that. Okay, so this is the D-aspartate, because if we want to read corn, we go from CO here to R to N, and we'll go around in the anti-clockwise direction like so, and hence this is the dextrorotatory enantiomer. So that's why the NMDA receptors are called the NMDA receptors. They're named after this agonist, which is a selective agonist for the GLU-N2 subunits, which are the glutamate-sensitive subunits of NMDA receptors. And I will stress this once again, that most NMDA receptors do have at least one, if not two, GLU-N2 subunits. Okay, right. So that now concludes our discussion of the ionotropic glutamate receptors.